genetic history of men, or more about what we have learned from human Y chromosome. And first, we talk a little bit about the evolution and structure of the Y chromosome itself. And then we look on the global phylogeny and variation with high resolution Y chromosome sequencing. And first, let's talk about the evolution of Y chromosome. Just very briefly, it is the male sex determining chromosome. So all men, mammalian men, have it. And the sex determining gene is located on the Y chromosome. But throughout the, when we go back in the history, it has been now said that, oh wow, sorry. It has been said that initially there were just 2 pairs of autosomal chromosomes about 300 million years ago. One of them then acquired the sex determining gene. And after that, the region which was around this sex determining gene stopped recombining. It went through inversion. So it did not recombine with its autosomal pair. And throughout the history of Y chromosome, what we see is this a sequence of inversions and rearrangements, and then also it is usually followed by the loss of genetic information in the Y chromosome. So in this slide, what you see is wherever there are these arrows, it means that there has been an inversion, and when there are big minuses, it means that there was a loss of genetic material, except in one place through the history of Y and X chromosome, which are the counterparts in the genome. There, around 150 million years ago, the other pair of autosomes infused with Y and X chromosomes, so they both gained a region which is called Y autosomal added region and X autosomal added region. And then from there on, Y chromosome continued to go through inversions and the loss, sequential loss of genetic material, until here on this very right of the slide, you can see that the Y chromosome is much, much smaller than the X chromosome. And this is now the human X and Y chromosome. And what happened after humans separated from old world or from chimpanzees is another addition to human Y chromosome, and that was a part of X chromosome that was copied into Y chromosome. So this is the latest addition to human Y chromosome. And when we now look at the Y chromosome in human whole genome context, then we see that it is very tiny compared to other chromosomes. It's one of the smallest one. It only takes up 2% of the whole genome. And it's about 60 million megabases long. And when we now zoom in to the Y chromosome structure, we can see that it is very particular in the sense that it recombines only in the very, very ends of the chromosome, which are marked with the green here. These are called pseudo-autosomal regions. So this is where Y chromosome and X chromosome recombine during the meiosis. And the great bulk of the Y chromosome does not go through recombination. And this is called the MSY on this graph. And it means male specific Y. And when we now would look into the different sequence categories or in the Y chromosome, one particular characteristic is also that there is a very large part, up to 40 megabases, which is heterochromatic. That means it does not produce any no transcription is from that region. So it's very densely packed. And the other parts which are marked with different colors here, I already mentioned the green parts, which were the pseudoautosomal. Then this pink part, which is called X transposed. So this is now the region, the latest addition to the human Y chromosome. And this part is most similar to the X chromosome by its primary sequence. And then there is the yellow parts, which are called X degenerate regions. And these are the oldest strata in the Y chromosome. These are the ones from this initial autosome, which then now has changed so much. And this has the least similarity to X chromosome. And also then Y chromosome is very particular about that it has very large regions of repetitive sequence, or which called ampliconic sequence, which means that there are actually sequences which are within the Y chromosome very, very similar to each other. There are big repeats, inverted repeats, and all this blue area is this repetitive region. So there is big, very, very big sequence similarity within Y chromosome. So actually there is recombination going on within Y chromosome. So Y chromosome exchanges material within itself, but not with X chromosome within this male-specific region. And when the initial Y chromosome sequence was published, that was already in 2003, then this was the comment about the effort that researchers had gone through to actually sequence this chromosome because of its very complicated structure and underlying sequence. So that there are lots of duplications, inversions, and otherwise troublesome sequences. So they were compared to explorers who had gone to fourth seas. So what can we do with Y chromosome? So you probably already know that, but I will repeat that we can study DNA in general, or genomes, from the living people and also people who died long time ago, so from the archaeological specimen, and this is called the ancient DNA. And when we look and get the DNA from the different populations across the world, we can do some population genetics with it. So what can we do? We can look into the lineages within a population, how they change through time, how their frequency changes. So when time goes this way, lineages change in their frequency, and they can do that just by chance, or then there are some forces which change these frequencies. We can see that sometimes populations go through bottlenecks, which means that there was before much more diversity within population, but then some kind of event happens, which after which only a small portion of the population carries on, and the new population has much less diversity compared to the initial one. Where similar effect can be seen in the founder event, when there is an initial diverse large population, and then smaller groups from this population are the founders of new populations, and usually they also carry only the small amount or a subset of the initial diversity that is present. And then how we present this data, we can show frequency of different genetic variants and show their frequency on a geographic map, or the changes of frequency according to geography. From Y chromosome, we can build phylogenetic trees, and also one can build networks. And also the shape of those trees and networks tell something about the underlying populations. And this all, of course, when we have done a very good sampling of a representative sampling of a population. For example, here there is one population with much more diversity, whereas the other one has much less lineages, and some of them have gone much higher frequency. So Y chromosome is one of those very handy tools, in the sense that it has a very particular inheritance. It is only inherited through a paternal line. That means that Y chromosome is given from fathers to sons only. And because it does not recombine, as it is, or the majority of it doesn't recombine with its counterpart X chromosome. So this male-specific part of Y chromosome is passed on as a single locus. And this means that it is gaining mutations. As it gains mutations through history, we can build true phylogenetic trees if we somehow can detect the commonalities between different lineages. So if a Y chromosome is passed down from a male lineage, some of those male lineages never reach today, the others reach. And knowing the common variance between those lineages, we can build phylogenetic trees. And this study has been going on already from 1990s, so for a long time. Initially, people were able to detect a few mutations, and as their research continued, the mutation databases were constantly improved. And Y chromosome researchers came together and established a certain rule how to call the different lineages. So bigger groups which have a common variant defining the bigger group, they were called haplogroups. So these are monophyletic clusters, or also could be imagined as lineages which have many sublineages. And then within those clusters, which were given alphabetic name, just letters from the alphabet, the subclusters were named with number and letter combinations. And also every node was defined by defining mutation. So lots of researchers did their research. They started genotyping many samples in very many populations across the world. That meant then searching for those common variants between individuals, and then building phylogenetic trees according to that information. And what was established was that male lineages have a certain geographic pattern, or they are not just randomly distributed across the world, just completely randomly. They seem to be that some of the lineages which here are marked, this image is taken from Wikipedia, but it just looks very nice. It's not definitely not the most up to date, but it still has the main information And what we can see is that there are these numbers, letters and number combinations, or the haplogroups, they have a certain distribution, geographic distribution. And when the phylogeny is built and the haplogroups were named, as I said, according to the alphabet, starting from A as the deepest branch. And another peculiar thing, or very characteristic, is that the deepest splits, or the very first splits in the whole human paternal lineage, have phylogeny. So in essence, you could relate all the men on the world through this phylogeny. So the very first splits are all within the African continent. So these, and the rest of the world, all the lineages which are outside of Africa, they are a subset or a subcluster within the African lineages. And this gave another proof to the out of Africa theory as well, or people migrating out of Africa. And there has been lots of research relating the phylogenetic trees to geography. As I said, it's called phylogeography. And it is presumed that when a lineage that is nested, or this would be the ancestral lineage and the derived lineages, and if they have a particular geographic distribution, one can say that can deduce the direction of the migrations as well from there. So this all has been done for many years. Lots has been established by this genotyping method. But now we have a whole new alley, which is sequencing Y chromosome. And this, you can imagine, I didn't write it out all here, but when you would sequence all the Y chromosome, then you have so much more information. So the phylogenetic trees are much higher resolution, or we can differentiate between the ancestral and derived lineages. When on this tree, we cannot say if any of those three lineages, for example, are somehow or which of them are closer related or which not. Then when we have more information, we discover more mutations and see that, let's say, these two lineages here share some mutations. We can draw a tree which then connects them to, for example. So by sequencing, we can gain so much more information. And one important aspect to this is that we can add time to the phylogenetic tree directly. There was also, and still is in use, another way of calculating time or divergent time, or time from when the lineages split from each other. That was based on STRs, but I'm not going to talk about this today. So because now we can sequence and we can construct the tree, and by counting the mutations, we can see that which of those branches have gone through longer time. When this one has only 4 mutations, it is much shorter in time than this one with 9 mutations, whereas this lineage, we haven't discovered any of the lineages yet, which would, for example, maybe share only 10 here and split somewhere. So importantly, we can add a time aspect with lots of sequences. And this has been very, very tightly linked to another concept, which is the mutation rate. There are several ways of calculating it. And one way is to calculate or calibrate the chromosome Y mutation rate from comparing the chromosome with ancient DNA. And very, very simply, I tried to explain is that when we have an ancient sample or a sample which age we know from what time this sample is. Let's say this is from an archaeological specimen from 6,000 years ago. And we have mutations which connected here to the other lineages, which are contemporary lineages or from the people living today. So when we compare the mutations that are missing in the ancient lineage and divide it with the age of the ancient sample, then we, in essence, can calculate the mutation rate for the Y chromosome. And this is one example here, because there have been quite few or several mutation rates for Y chromosome. Now they are similar, quite similar to this one. I'm just presenting this one here because this is the one that we used for the results that I will show just now. But another word about, or like a word of caution about the sequencing technologies. If you remember, I was telling that the Y chromosome sequence is very complicated, that it has lots of inversions, repeats within Y chromosome. Then the most common sequencing method today, which probably you have heard many times now, but just to repeat that it creates very many short sequencing reads, very many short pieces of the genome. There is also two ways of getting a Y chromosome sequence. One is when somebody sequences whole genome of a man or many men, and then the whole genome is shared into small pieces, and these small pieces are amplified and sequenced. Another way is targeting certain small regions already within Y chromosome. This is called Y chromosome capture. Both of those ways, or both of this data is used today for Y chromosome studies. So when these small amplified regions go through sequencing platform, this produces a myriad of small reads, which are basically from anywhere from the genome or then from different parts of Y chromosome, but they are all mixed together. And the mapping process is very important. In case of human genome, we are very lucky because we have a very good reference genome. There has been lots of work done, so the reference genome would be as accurate as possible. The work is ongoing because still there are improvements that can be made, especially in regions like Y chromosome, also X chromosome, which have also many regions which are complicated. So, but still, Y chromosome has lots of repetitive sequence, so it is hard to tell from if there is sequence which is similar in one place of Y chromosome to a sequence in another place in Y chromosome. So when we are mapping, we don't know from which region it comes from. Therefore, we can use only small part of Y chromosome sequence, and this is the most unique part. And it is around 10 megabases, a little bit more, a little bit less. It depends on a certain on the specific study and specific methods that people are using. So now, when talking about the results, so that would be about the phylogeny and some population dynamics of Y chromosome based on sequences. So already 5 years ago, we had I had a great pleasure of working with many great people on this project, which sequenced or used sequences from 459 men across the world, from all different regions of the world. And we were able to capture 8.8 megabases of chromosome Y sequence, which when we think about the whole chromosome being 60 megabases is not maybe so much, but comparing to whatever was available before, it is so much more data. And these men came from 128 populations, and they are colored with these different colors, which are listed here. And what we did, we built the Y-chromosome phylogeny, and we were also very lucky to have the or to be able to include the oldest human lineage that had been detected, or this is still the oldest lineage detected up to now. So we were able to root the tree, and all male lineages in among humans coalesced 254 kiloyears ago. I say 54, it's let's say it's about 250,000 years ago, because if we'd use a little bit different mutation rate, well, the 54 wouldn't be, maybe it's a bit too accurate, but in that ballpark. And also we saw the same, that the deepest lineages deepest splits are the ones, the first ones, which first splits, these are all within Africa. And then the lineages which are outside Africa are all those lineages which are here marked with GT and C. Also, a haplogroup E is found outside Africa, but these two are like purely only outside Africa. And they split from the African lineages, but then they go through about 15,000 years incubation before all the African lineages suddenly go through great expansion event. You see how, I go back now, you see how here there are just these longer branches and no splits, whereas suddenly in this time period you see how all the main lineages, which now are across the globe, from Europe to Oceania and also to Americas, they are all the main lineages appear in this very short period of time. So there is a sudden expansion of lineages. And this is the time frame that also, in accordance with archaeological findings, we already know that Australia was already inhabited by people by that time. So it is a great expansion of human lineages. And then another very broad characteristic that we see from this tree is that there are very many expansions happening also in the past 15,000 years. And this is the time when the climate improved after the last glacial maximum. So we see globally also that there is great expansion of lineages. When we would look more detailed into some regions, then there are lots of very many regional events also going on. But now here we are looking at the very global human paternal phylogeny, and it is happening everywhere. And another thing that is possible to do when you have sequences is the estimation of effective population size. This is just a reminder that what is effective population size? So very broadly said, it is that the number of breeding individuals. It has also different definitions. But if you think that the number of actual individuals in the population does not usually represent the number of breeding individuals, or those who actually give their genetic material on to the next generation. And there is differences between male and female effective population size. This has been studied also for a long time. And it's been said that there is an unequal variance in the number of offsprings, which in other words means that there are some lineages or some men or some women which are more prone to have offsprings than other women and men at the same time. And it has to happen also sequentially through the generations, so the effect would go on. And just a reminder also that there is a relative effective size is different between autosomes. Effective population size is different between autosomes, sex chromosomes, and mitochondrial DNA. But this difference that we are talking here is on top of this relative difference, which is there because if we take a man and a woman, they have altogether 4 autosomal chromosomes, 3 X chromosome, only 1 Y chromosome, and only 1 mitochondrial DNA. And it has been also studied that how do cultural practices influence genetic diversity, because it is clear that when the effective population size is smaller, there is less diversity. It means that less lineages are carried on to next generations. And one of the well-studied cultural practices, matrilocality and patrilocality, and this it has been shown that in general, in human populations, within the differences, genetic differences between different populations of males is usually bigger than that of females. And this is because for a long time now, human populations have more been patrilocal, so the males have stayed in one place where females have moved rather to their partners. And it also has been shown that cultures where matrilocality has been practiced, the female differences between populations, which means also differences within populations smaller and between populations bigger. So this is something that was studied before, but when we were able to sequence the Y chromosome and take the mitochondrial DNA lineages from exactly the same people, so these lineages, and then construct the phylogenetic tree, and then there are ways to calculate the dynamics of effective population size through time. So on this figure, the time moves from left to right, and there is the effective population size is shown on the vertical axis, and this part is about Y chromosome, and this part is about mitochondrial DNA. And as I said, they are the mitochondria and Y chromosomes from the same individuals, so they represent these people's paternal ancestry and maternal ancestry. And already when you look at the tree, I guess you can already see the differences between the topologies of the tree just by looking. You see there are these lines which are drawn to 50,000 years ago and 10,000 years ago. When we look at the maternal side, then we see that there is much more coalescence is happening between the 10,000 and 50,000 years ago. And of course, as we move towards presence, there is always more coalescence is happening. This is already said by theory that this is how it works, but when you compare to the male side, then you see that there is much less splits happening in the male side of the tree. And when this effective population size was estimated, then very strange effect appeared. When you look at the male side, it looks like a snail. It has a drastic decrease here. Around between 8 and 4,000 years ago, there is a drastic decrease of effective population size of males compared to the females. There is nothing similar happening at the same time, and it is happening basically in all geographic regions. On this plot, the colors are marking these different regions. We see that it is not so prevalent in Africa, but in some other places, and most of the other places, it is very systematically happening around the same time, and nothing similar in the female side. So we were trying to see in many ways how to that it's not some kind of artifact of our results, but we couldn't figure out any way that something has gone wrong. So this is the case that in the male lineages, there is a bottleneck between 8,000 and 4,000 years ago. And when we zoom in to that particular part of the plot and now try to separate these different regions, so this is a little bit different plot, but it is showing the same thing. This is the time here, and this is the relative effective population size, and it has been normalized to these values 12,000 years ago. And then when looking at the time sequence, how this dip or decrease in the effective population size is happening, it struck the eye that it is broadly following the timing of when agriculture or Neolithic revolution had already been established in these different regions. So here again, these different colors are marking different regions. For example, this green is South Asia, Near East with orange. And when we look at the timings when the agriculture had already been established in these regions, it definitely came later to agricultural revolution reached Europe later than Near East. So, and we see the same pattern here among those effective population size decreases, that it is going in the same sequence. So we hypothesized at the time of the study that there could be something that everybody is sure that there has to be some kind of a force that is making these Y lineages disappear, so to say, or why is it that some men are having more offsprings than others, and this is also continued through generations. So their Y chromosome is passed on through generations, whereas other men, their Y is not. So we hypothesized that it could be something culturally inherited. So it's a culturally inherited variance of male reproductive success, which means that some have men have and their sons are able to pass on their Y chromosome, where others are not. And this is, of course, a hypothesis, so we couldn't test it anyway at the time. But this is just a graphical, very summary of this, that there was a rise of agriculture, and then we see that there was an extreme reduction in male genetic diversity and nothing similar in the female population. Actually, the difference between the most extreme difference between male and female effective population size here was 17 times bigger female effective population size than male. So we had, this is very simply said, so to say, survival of the wealthiest, but it could be something else also. So it's always interesting to hear also what people think, what could be the reason. And all this kind of information does have some health implications, that it means that we have detected time in the history when there has been a reduction in genetic diversity. And this same effect has been shown in other studies as well. Here is another study, but with very similar plots, showing all these different regions where the red is for mitochondrial DNA, and these two blue on each of those plots are marking Y chromosome. And basically the decrease of effective population size is also visible here on those plots, except in some regions, which are also not very well covered by samples, unfortunately. And another study showed, which was later study, also based on sequencing, this is 1,000 genome samples, showed how these in different regions of the world, there have been certain male lineages which have gone through great expansions. So within Africa, there is the haplogroup E lineages, which are now very widely distributed also among Bantu-speaking people. But this expansion was before the actual great population movement within Africa. So the time again is here, the timeline. So that was around predating this big migration. So the authors of this paper also speculated that what could be the actual causes. So they speculated that this could be somehow related to the iron working that started to be very common at this time. So this led to some men being very more effective than others in giving offspring. When now we're jumping a little bit in time, but the next one in the graph is Americas. So this is much further back in time, 15,000 years ago. And the haplogroup Q, which is the founding haplogroup. And this is exactly the time when the archaeological evidence also points to the first humans actually expanding within this continent, and they carried this one of the main male lineages. So it went also through great expansion there. In East Asia, there isn't such big sudden bursts of or expansions going on, and it is also maybe because the initial population sizes were actually already, to start with, they were much larger. But still they could detect two expansions within haplogroup O. One is 8,000 years ago, another one 4. In Europe, very prominent expansion, which is also related to several archaeological cultures. And then also in South Asia, there is altogether 3 haplogroups which show this kind of expansion, but there definitely should be more ancient DNA to relate those to specific events. So ancient DNA, ancient DNA has been very helpful, and also sometimes refuting the earlier theories about which have been put forward only based on the contemporary individuals' Y chromosomes. There was another study which came later, and it was a modeling study. It's a very interesting study. I really recommend to read that. And which was trying to see what could cause such bottlenecks in the male effective population size. And they proposed that it is patrilinear kin groups, and then there is an intergroup competition. Kin group means that basically one male lineage is the main lineage within the group, and when there is competition, it means that some other lineages are killed off, and then those do not kill any more offsprings, and therefore there is a reduction in the diversity of Y lineages. They also showed this how the different cultural well, these are all from the archaeological findings or the ancient DNA results from other studies, and how the Y chromosomes from the same sites in the same archaeological culture, from hunters and gatherers, they coalesce much further back in history than, for example, when we take these samples here, then samples from different locations are having a very, very shallow coalescence. So they have a very recent common ancestor, again showing how the differences in, and these are from the agriculturalist already. So they showed how the populations who were practicing agriculture and those who were hunting and gathering, and then the pastoralists also, they all, agriculturalists and pastoralists, have much shallower Y chromosome trees, or their common paternal ancestor is much younger than for the hunter-gatherers. And I think it's maybe a good time to stop here now. So for the very, very broad conclusions from this global phylogeny, it coalesces about 250,000 years ago. And the main deepest splits are within Africa, and all non-African lineages differentiate in a very short time interval, around 50,000 years ago. And also that within the past 15,000 years, there are many, many expansions or punctuated bursts, as they were called. And another interesting feature, which has been now recently discovered, thanks to the sequencing capability that we have now, that there was a decline in the male effective population size 4,000 to 8,000 years ago, and possibly it has cultural causes, or it was culturally driven, and the force was driving the increase in the variance of male fitness, which means in essence that some men could pass on their Y, that there was some male lineages which were favored over the others. And these have been the explanations that the researchers now have been also proposing for this. And thank you. That's the end.